one of these, you may be asked to describe what category of end-linked glycoprotein you have there. So take a look at this. In my opinion, the easiest one to identify is high mannose because it's in the name. It just has a bunch of mannose sugars in it, which was one of the monosaccharides you had to memorize earlier. Good. In 8A, we learned about all those different types of lipids, right? Fatty acids, triglycerides, phospholipids, glycerophospholipids, plasmalogens, sphingolipids, cholesterol, blah, 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 blah. Most important ones you need to know, starting, is this particular one called, I can't pronounce it, CHA. We learned about this one in detail, and you were asked, I believe you were given something, a question involving this one in exam 2 that was asking you to look at the structure of this thing and what's true or false about it. So it is still important for the final exam, so please review it. There's some features listed here about it, so make sure you know them. You might remember that there's a notation we use to identify unsaturated fatty acids. And that's 3 numbers. The first number in the notation tells you how many carbons a particular unsaturated fatty acid has. The second number tells you how many double bonds that a particular unsaturated fatty acid has. And can anyone tell me what the third number tells you? Something called the omega number of a particular unsaturated fatty acid. If you guys don't remember what omega number is, I'll illustrate that with this one just for practice. Every unsaturated fatty acid has an omega number. You can identify what value the omega number has by starting at the very end of that unsaturated fatty acid and counting how many carbons it takes to get to the first double bond that you run into. So for an example, this one has an omega number of what? 3, because you go carbon 1, carbon 2, carbon 3. That ends up being the omega number for this particular fatty acid. All right, continuing right along. Okay, some fun stuff. There's a category of lipids called sphingolipids. All right, there's a category of lipids called sphingolipids. They're derived from a base structure known as a sphingosine. So let me pull up a sphingosine for you. That's this one right here. Probably important. This sphingosine is slightly modified into something called a ceramide. And this thing over here on the right, you can attach various different things to it, and you get different types of sphingolipids. Some examples include, you take a ceramide, and you attach a polar phosphate head to it, you end up creating sphingomyelin, which is important for what types of tissues in the body. Neurons, nervous tissue, has lots of sphingomyelin in it. It's a type of sphingolipid. All right. There are other types of sphingolipids that are discussed. This one is a ceramide with a single sugar attached to it. We use the vocab word cerebroside to describe that. This one's the easiest one. You have a ceramide attached to many sugars. This is a ganglioside. Okay, a gang of sugars is how I remember it. So those are your sphingolipids.
Know the structures, right?
Yep, know the structures. In particular, there's one disease that was discussed in this class regarding these sphingolipids. Some of you might remember. I will highlight it again here. This one, Tay-Sachs disease. Make sure you review the Information given about Tay-Sachs disease that might be asked about. That is in the PowerPoints there. Everything I'm discussing is in the PowerPoints, and we reviewed it at SI. Chapter 8b, also known as Chapter 9, talks about the different types of membrane transport systems. You have passive transport and active transport. You have symport, antiport, uniport, and you have very specific pumps that were discussed and transport systems that were discussed. So let's point out a few of them. We have a type of transport called passive transport, of which there's a specific category called facilitated diffusion. You might remember facilitated diffusion. The way this works is molecules travel with the concentration gradient. They travel with the concentration gradient in facilitated diffusion. However, interestingly, they do so via membrane transport proteins. So they don't do it by themselves, they do it with membrane transport proteins. Because they go with the concentration gradient, no energy is used. You don't need ATP, I should say, to power any of these things. A good example of a facilitated diffusion system is the sodium glucose symport. The sodium glucose symport. Okay? This thing can be found in both the intestines and the kidney, and the way this works is, I'll use the intestines to describe this. When you ingest chocolate, or any type of sugar, it enters your body, it enters the stomach, it's broken down, it's in the small intestines. It enters the intestinal cells by pairing up with sodium. That's where the name sodium glucose symport comes from. It pairs up with sodium, goes to this transporter, and gets moved into the cell. Afterwards, the glucose will eventually be moved from the cell into the bloodstream through a secondary facilitated transport. All right, but the main important part is the sodium glucose transporter. It's the facilitated diffusion, does not require ATP. I know you might see that, oh, sodium is ATP transported. That does not mean this pump is ATP dependent. This phrase here just means that the sodium got outside of the intestinal cells through a different mechanism. Specifically for the context of the sodium glucose symport, it is facilitated diffusion. So especially pay attention to the fact that glucose needs sodium to be transported into the intestinal cell. Especially focus on that. Lastly, we learned about one drug associated with this symport called, I believe it's pronounced Farxiga. Forgive me if it's mispronounced. Farxiga. This particular one inhibits the sodium glucose transporter in the kidneys. Okay, it inhibits the sodium glucose transporter in the kidneys. In part, this is used to help treat high sugar content in the blood, to help treat diabetes, to help treat high blood sugar using this thing. And it inhibits the sodium glucose symport in the kidneys. And it inhibits the ability for kidneys to reabsorb glucose. Okay. The most important active transport system you should know about is the sodium-potassium pump. Okay? The sodium-potassium pump is very simple. It pumps sodium against its concentration gradient and potassium against its concentration gradient. Visually, that is described as, this pump takes 3 sodiums at a time and throws it out of the cell. In response, it grabs onto 2 potassiums that are outside the cell and moves them inside. Because these are both against their concentration gradients, it requires the hydrolysis of ATP to power this thing. Which side of the cell, intracellular or extracellular, is the only place you can hydrolyze ATP? inside the cell, intracellular. You never have ATP hydrolysis outside the cell. That's not where you find ATP. ATP is always inside the cell. Therefore, if you want to hydrolyze it, if you want to build ATP, do anything with ATP, it is always intracellular. And again, pay attention to the sodium-potassium pump. What directions these ions go in when this pump is working. Okay. That covers 50% of the exam. 50% of the exam is exam 1 and exam 2. The remaining 50% of the exam is exam 3 material by itself, plus 29B, 30A, and 30B. So that's what we're going to start on now. So exam 3 material, plus the other stuff after exam 3. We will begin with the structures of your nitrogenous bases. Okay, you need to know the structures of your nitrogenous bases. A lot of this probably sounds the most familiar because we just went over it like a couple weeks ago. So make sure you know what cytosine looks like, uracil, thymine, guanine, and adenine. And again, because this is a multiple choice exam, just know them enough that you can pick one from the other. And I gave you shortcuts on how to do that. You all might have come up with your own shortcuts as well. Make sure you know your nitrogenous bases. Oh, and of course, know which ones are found in DNA and know which ones are found in RNA. I know that goes without saying, but just in case, I will reiterate that. There's one nitrogenous base in here that's a little annoying because it can spontaneously transform through a deamination and hydrolysis reaction into uracil. That would be this nitrogenous base, cytosine. Cytosine can spontaneously transform into uracil. That happens a lot. You may not be given a question that simply asks that. You may be given a question where knowing that information will help you greatly in solving the question. Okay? So make sure you know that cytosine sometimes transforms into uracil. Okay. You saw this on exam 3. There was like 1 or 2 questions asking this, where they give you the percentages of A's, T's, G's, and C's in single-stranded DNA, where they give you the percentages of A's, T's, G's, and C's in single-stranded DNA, and they want to know the percentage of a base in double-stranded DNA. I talked about how to solve that, right? You calculate, you draw out your DNA, you base pair everything, and you add all the percentages up that you need to, and you divide it by 2. You don't know what that means? Go look at the exam 3 review stuff. Again, you may be asked to review, you may be given information about single-stranded DNA, the percentages of bases in single-stranded DNA, and you need to find out something about double-stranded DNA. Okay. I can't remember, this is at the end of, oh yeah, no history thing, never any history thing. Don't worry about, like, when something was discovered, don't worry about that. Okay. We'll use 10B to talk about the next thing. And that is the phrases polycistronic mRNA and monocistronic mRNA. They're slightly different from each other. They are, you have compared and contrasted them for a while now. You may be asked to do that again for the final exam. Some things to watch out for, and it may not be everything. Some things to watch out for are polycistronic mRNA are in prokaryotes, so only prokaryotes polycistronic. Polycistronic mRNA has multiple genes on it. That's why we call it polycistronic. It's one mRNA with multiple genes encoded on it. To contrast that, oh, and the word we use to describe that is contiguous, which is listed here. There's another type of mRNA called monocistronic mRNA. Monocistronic mRNA is only a eukaryotic thing. And the reason why we call it monocistronic is because it's one mRNA for one product. So a single mRNA is only going to make one single product. Other features of monocistronic genes are the fact that they come from genes containing introns and exons. You guys need to remember what the introns and exons are. Exons are your protein coding sequences. Introns are the useless stuff, oversimplifying it, are the useless stuff that gets spliced out during transcription. There are a couple other modifications we do to monocistronic mRNA in eukaryotes. There are two modifications we do to it after splicing. We attach what to the tail? A poly-A tail, and we attach what to the head? A 5' G cap. Those are key features of eukaryotic mRNA that prokaryotic mRNA does not have. And I'll talk about it here since we're on this topic. Why can't we take prokaryotic mRNA just as it comes, throw it into a eukaryote, and why can't eukaryotes transcribe it? What feature is missing on prokaryotic mRNA? The 5' G cap. I'm telling you now that that is going to be the concept for the final exam. I sent an email about it, so you guys already know this. I will reiterate it here though. The 5' G cap, because it's missing on prokaryotic mRNA, the eukaryote does not understand how to translate that prokaryotic mRNA because of that. Thus, if we want to do any experiments where we take prokaryote and throw it into a eukaryote, we need to attach that 5' G cap to a prokaryotic mRNA. Okay. I may be going a little bit out of order here, so forgive me if I'm jumping around. These things, your exonucleases, right? There's 4 combinations here, 5'A, 5'B, 3'A, 3'B. You may be given a DNA sequence, you may be given one of the 4 possible exonucleases, and just like for exam 3, you may need to find out what fragments you get when one of those exonucleases are applied, all right? So make sure you review how to do that. I've gone over that before. Again, how to using an exonuclease on a given DNA sequence to figure out what fragments of DNA, and you must figure out what fragments of DNA you will get from that. MicroRNAs, there's lots of them, don't worry about memorizing them. Just the general feature of microRNAs that you learn in this class is that they're heavily involved in embryonic development. When an organism is in development, it uses microRNAs to regulate transcription and the expression of genes. MicroRNAs are critical for the development of organisms, for the development of embryos. Okay. All right. After exonucleases, there's a category of enzymes called endonucleases, a synonym of which is restriction enzymes. You saw some questions about restriction enzymes on exam 3. You may get additional questions, such as this thing. I did promise that if it wasn't on exam 3, it'll show up on the final exam. I have detailed this enormously at SI, and not for the exam review actually. So this one you're going to have to go look at the SI videos for this particular topic. It is in the chapter 10 SI videos near the end. Make sure you look at how to solve a recombinase cloning type question. Yes?
That's the one that has like the gene
and the circle. Yes, exactly that one. You have a circular plasmid. You may be given a gene and a list of restriction enzymes, and you may be asked, which one can you use to clone the gene into the plasmid? All right. There is also, I believe, one practice exam question that's like that that you can review as well. This next feature, actually it is right here. There's one feature of restriction enzymes I forgot to talk about. So if you don't remember, restriction enzymes, their job is to cut DNA at particular sites in a genome. bacteria actually naturally use restriction enzymes as a defensive mechanism. Think of restriction enzymes as kind of like a bacteria's immune system against foreign bits of DNA and RNA from like viruses. Bacteria use restriction enzymes to cut up these foreign bits of DNA and RNA. But you might wonder, well then how do bacteria restriction enzymes not avoid accidentally cutting themselves, cutting bacterial DNA? And that is because of this neat feature here. Bacterial DNA has methyl groups on it, That is kind of an identifier for restriction enzymes to leave those bits of DNA alone. It's kind of like a self-identifier for restriction enzymes in bacteria. Methylated DNA communicates to restriction enzymes not to cut it. So that's how bacteria does not end up hurting itself with restriction enzymes. Foreign DNA and foreign RNA from viruses do not have these methyl groups on there, so restriction enzymes are still able to cut those. Okay. Decided to simplify this type of concept. This is supercoiling and linking number. Linking number, the formula is L equals T plus W. If you're given any combination of two of these numbers, make sure you know how to calculate the third one. So simple algebra. Make sure you know the formula though, because we are not going to give you the formula. Another simplified thing on here, and I have mentioned this, is if you have relaxed DNA, not supercoiled DNA, unsupercoiled DNA and relaxed DNA, W equals what? 0. All right? Two simple facts that allow you to answer most supercoiling questions. Cruciform. There was a question on exam 3, you might remember, where given 4 different sequences, you had to identify which one forms a strong cruciform. You might remember that you have to look for 2 things in that case. The first is you find which ones display something called an inverted repeat, which I've described before as you just read the sequence from left to right up here, and you go to the bottom right end and read it in the opposite direction. If they are the same pattern, that is an inverted repeat. Structurally what happens is that it ends up binding to itself. Each individual DNA strand ends up binding to itself rather than the opposing DNA strand. A strong cruciform, you have to look for one additional thing, which is what? C's and G's. A strong cruciform is one in which also it must have lots of C's and G's in it, because we know that's a strong type of hydrogen bond between nucleotides. I didn't pass it. Now, here we go. Chain termination method. You saw it on exam 3. You might see it on the final exam again. You may be given a gel. It'll look nicer than exam 3. You may be given a gel. You may be given a primer. Same thing as on exam 3 you're going to have to do here. Find out what the original DNA sequence was. Okay? So if you're given a gel, use the information we learned before to sequence the original DNA sequence. Okay? God, there's so much. So, going back to restriction enzymes briefly, restriction enzymes by definition are able to cut particular patterns that are found in DNA. Because of this, we can actually use restriction enzymes to identify differences between one person's DNA pattern and another person's DNA pattern. You know, those types of forensics things that you see on those detective shows. Biologically in the lab, what we do with that, and there isn't a nice example illustrating this, but we take two people's DNA, we throw a restriction enzyme on them, it chops it up into little fragments, and you can run gels on both of their samples of DNA. When you compare their gels to each other, when you compare what bands show up where to each other, you can compare that to a DNA sample you would find at the crime scene. A DNA sample you find at the crime scene will cut in the exact same pattern as a later DNA sample taken from the person who committed the crime. So it's a common type of story Dr. Hayes probably went over during class. I'm just re-illustrating it. You can use restriction enzymes to compare genomes to each other. It's the biological reasoning for this. You can use restriction enzymes to compare genomes to each other. So imagine you have a lot longer of a gel than this with like tens of bands on it. You can compare person A to person B to person C to each other by how many similar bands they have in that gel. Another application of this is comparing family members to each other. Two people that are very related, that are closely related to each other, let's say parent and child, will look very similar in their gels when we use restriction enzymes. Two people that are not related to each other, when you look at their gels, the bands should show up in completely different places. So again, sounds annoying, but it is an application of restriction enzymes. It is forensics, family lineages, things like that. Okay. Pay close attention, especially when we're talking about that DNA sequencing thing, that when you do this, pay close attention to the direction of DNA that you write down. A common mistake that some people brought up to me is they would sequence the DNA out of this gel, but they would write down the wrong direction, or they pick the wrong answer choice because it's in the wrong direction. So make sure you're very carefully writing down the directions, the correct directions, like 5' side and 3' side whenever you do these things. I've gone over a lot of that during the reviews and during SI sessions, so pay special attention to that because I don't want anyone making those mistakes for the final exam. Alright, DNA replication. Most of the information we wanted to ask you guys about DNA replication is actually on exam 3. However, there's a few things that you do need from here for the final exam. In particular, eukaryotic information. So, same as for exam 3, you need for the final exam is that eukaryotic replication is driven by several different DNA polymerases. DNA polymerase alpha, which is able to make its own primers. DNA polymerase beta, which is similar to alpha, but it's special for repairing DNA. DNA polymerase gamma, which is responsible for replicating DNA in mitochondria. DNA polymerase delta and epsilon, which generate most of the DNA in eukaryotes. Delta is the one that replicates DNA on the lagging strand, and epsilon is the one that replicates DNA on the leading strand. So features that have been brought to your attention before. Also unique for eukaryotic DNA replication is some cells have the ability to repair their telomeres. So what are telomeres? So eukaryotic DNA is linear. That comes with it some problems in the fact that every time DNA is replicated in eukaryotic cells, it will get shorter and shorter because of the following fact. Telomeres are normally unable to finish their own replication because of the lagging strand. The lagging strand, you're unable to finish replicating the lagging strand. Most cells end up losing their DNA a little bit at a time until they reach a limit called the Hayflick limit, which is DNA has shortened so much that it is unable to replicate again. Some cells, ones that replicate a lot, have the enzyme known as telomerase. Telomerase is a special enzyme because it's a reverse transcriptase, and the job of telomerase, you can visually see what it's doing here, is to extend the template lagging strand. That's all that telomerase's job is. It's a reverse transcriptase, and it's going to artificially extend the template lagging strand. Now that you have more lagging strands, this enzyme, DNA polymerase alpha, can finish replicating the lagging strand. So both of these put together are necessary for replicating DNA in telomeres. Not all cells have telomerase. Only cells that replicate very quickly have telomerase. And you may remember a question about that on exam 3. All right. Okay. Going to reiterate some things because some people had questions about this. This is 29A, this is transcription. We use vocab words such as sense and antisense. And now you can see on this slide what each one means. The sense strand of DNA can also be called your non-template strand of DNA. Take a look at the mRNA that was made, and you can compare it to the sense strand of DNA. What do you notice? You should notice that the sense strand of DNA and the mRNA that is made in transcription look very, very similar to each other. They almost look exactly the same. So the sense strand of DNA, the non-template strand of DNA, looks very similar, almost exactly the same as the mRNA strand. There's just one difference. Every thymine that you see in the sense strand of DNA is replaced by what in mRNA? Uracil. Exactly. That's the only difference. Everything else in sense DNA you will find in mRNA. So, you saw an example question in exam 3 where you're given sense strand DNA and you were asked, what's the mRNA sequence that you get out of it? So pay close attention to that type of question. Just in case, read it carefully, because what happens if it says antisense DNA and you're asked to find mRNA instead? Make sure you remember that antisense is another word for template strand DNA. And I will remind you that warning I gave earlier, pay close attention to the directions that you write down when you draw matching base pairs to things. When you write down a template strand, pick a direction that's 5' and a direction that's 3'. And whatever complementary strand you are writing down, make sure it's anti-parallel to that template strand. Template something, and the thing that you are synthesizing must be anti-parallel to each other. Another warning again also is that if you're given any sequence, DNA or RNA, where directions are not labeled, you have to remember that the left side of an unlabeled sequence is what direction? 5'. And the right side of an unlabeled sequence is therefore 3'. Okay, so other warnings to give there. Okay, I will probably jump around here. This is a pretty big section. It doesn't mean you skip all this. I'm just looking for something. Okay, so it's not here. Hold on. All right, going back up. So, transcription. We learned in prokaryotes that all of transcription is taken care of by one enzyme called RNA polymerase. This RNA polymerase is made of several different subunits, alpha, beta, beta prime, and sigma. Pay special attention to beta, beta prime, and sigma. And those, the the features are listed on here. Beta prime binds DNA, beta binds new nucleotides, and sigma is the one that recognizes promoter sequences in transcription. This one RNA polymerase takes care of all types of RNA in prokaryotes. So this one polymerase will transcribe mRNA, will transcribe rRNA, and will transcribe tRNA. Contrast that with eukaryotic transcription, where you need a different RNA polymerase for mRNA, a different one for tRNA, and a different one for rRNA. Somewhere in your notes, they'll tell you which RNA polymerase is for which. You can find that in your notes. But for prokaryotes, a single one takes care of all three. When we talk about transcription, there are 3 steps: initiation, elongation, and termination. We asked about initiation a lot on exam 3. Therefore, for the final exam, we're going to be asking about elongation and termination. So let me jump to that. Elongation is pretty easy. The only information you need to remember about the elongation step in transcription is the fact when you transcribe something, you are opening up DNA. Whenever you open up DNA, you build up supercoiling. We learned there's a category of enzymes whose job is to fix supercoiling, of which there's one that fixes positive supercoiling ahead of the transcription bubble. What enzyme takes care of that? Gyrase. And there's a category of enzymes that fix supercoiling behind the transcription bubble called topoisomerase. And I think it's topoisomerase 1 specifically. When we talk about ending transcription in prokaryotes, there are 2 ways to do this. This wasn't talked about a lot for exam 3. This will be asked about on the final exam. The 2 ways are row-dependent termination and row-independent termination. So let me illustrate the 2. Firstly, row-dependent termination is the ending of transcription via the protein called Rho. Look at this slide and it'll illustrate exactly what's happening. The bottom line is this protein ends transcription by using its helicase activity to unwind everything out of the transcriptional bubble. To contrast this, row-independent termination does not use the Rho protein. Instead, it uses the sequences itself to end transcription in prokaryotes. The way this works is that an inverted repeat pattern, when it is transcribed, forms this shape here called a hairpin loop. This is a very destabilizing structure for the transcriptional bubble, and to oversimplify it, this hairpin loop is the reason why the RNA polymerase falls off the template DNA and you end transcription because of this. So those are the 2 ways prokaryotic transcription is terminated / prokaryotic transcription is ended. That is row-dependent and row-independent termination. Transcriptional regulation in prokaryotes. Lots of you did really well on the extra credit. That's good, because you may be given several multiple choice questions asking about the Lac Operon. Okay? We know there are 2 components to regulating the Lac Operon. Oh, I don't like these diagrams, but you know them. You've all drawn them for me. I've had to read all of your extra credit questions. The first way the Lac Operon is regulated is via what mechanism? Negative inducible, right? You may be given questions about that, such as what are the co-inducers of that? How does that work? What about the second part? What's the second way a Lac Operon is regulated? Positive inducible via an inducer called what? Cap, right? You may be given questions asking about that. You also even told me, lots of you told me on the extra credit, what happens when you have lots of lactose, lots of glucose, or not a lot of either of them, or one or the other. You told me what happens in each of those situations. You may be asked about that on the exam as well. So I expect nobody to get anything wrong regarding the Lac Operon. You guys did very well. You guys made me grade things all night long for that extra credit. So I expect everyone to do well on that. You are welcome.
John, you said that if we are asked how the Lac Operon is regulated, it's both negative and positive?
Yeah, good question actually. The Lac Operon, technically, if the question is only how is the Lac Operon regulated, technically the answer is the negative inducible part, because the cap stuff is secondary, that is bonus stuff, okay? So if it's asked about how the Lac Operon